name is Sierra Bruni, and this is my audience.
Hello, my name is Sierra Bruni, and today I would like to start by asking a simple question. What if we could see memories? Now we all know that memories are stored in the brain, but what if you could open up the brain, look inside, and see the memories it contains? Well, that's what I'd like to talk about today. I'd like to explain how, using the catFISH method, we can actually look inside of a brain and see the memories inside. But before I do that, I should give a little bit of background information. The catFISH method was first developed in 1999 by Dr. John F. Gazowski. Later on in 2001, he formalized this method, and more recently, in 2019, Dr. catFISH used the method to investigate how memories change with age in mice. Also, just a few years ago, I used this method myself when working in undergraduate research. Another thing to know about our catFISH is that it allows you to not only see one memory in the brain, but actually two. And to explain how that works, I find it's easiest to explain what each letter in the ARC catFISH method means. So to start with, I'll explain ARC. ARC, you'll see here, I've represented in red. ARC is a protein. This protein has many functions that have been investigated by many different researchers. For example, Dr. McIntyre, who I work with in undergraduate research, she investigated ARC's role in learning in a broad sense. Other researchers, such as Dr. Huff and Dr. Monti, have investigated its role more narrowly in fear-based memories. But really, you don't need to know the function of ARC. For the purposes of the catFISH method, all that you have to know is that ARC is made in a cell immediately after it's activated. What this means is that when a memory is activated, such as when you recall the information contained in that memory, the cells that make up that memory all start to produce ARC. The next letter I should explain is C. C stands for cellular compartmental. This refers to the fact that in every cell, there are two compartments. One compartment is the nucleus, which I have labeled in these diagrams of cells with an N. The other compartment is the cytoplasm, which I've labeled with C. The next letter to explain is A, which refers to analysis. In this case, we are analyzing where ARC falls within these compartments to give us an idea about T, the temporal activity. This temporal activity, which refers to time activity or time-based activity, this refers to the fact that when these cells, after they're activated, when the memory that they form is activated, all of these cells will start to produce ARC, as I said, and where that ARC is located in the cell after that point is very time-dependent. So when the ARC is first produced, it is made in the nucleus, again, the central compartment. It stays there for about 5 minutes, and then after 26 minutes, it moves out into the cytoplasm. This means that just by looking at the cells and where the ARC is within the cells, we can get a sense of what memory that cell was involved in. If you activate a memory 5 minutes before performing ARC catFISH, then you will see the ARC is located in the nucleus, such as in cell 1. If you activate a memory 26 minutes before, then all of the cells that form that memory will have ARC in the cytoplasm, such as in cell 2. And if a cell is involved in both memories, it will look like cell 3, with ARC in both compartments. And if a cell is involved in neither memory, it will look like cell 4, which does not have any ARC present. In this way, we can actually see the memory based on how the cells that form that memory look. But there's another important part, which is the rest of the term, FISH. FISH stands for fluorescent in situ hybridization, which is a very big fancy word. But you're probably familiar with the term fluorescent, which just means to glow. And in this case, the glow is a red color. But the red is not actually ARC. The red is a marker that we use to mark ARC. It's a red fluorophore called Psi. Again, all you need to know is that it is a fluorescent molecule that looks red. And we attach attach it to ARC by a method called hybridization, and that allows us to make ARC look red. And that, again, together allows us to see what memories are stored in what neurons. So we can see the memory within the brain. And that overall is a brief introduction to the catFISH method. Hopefully you understand the method a bit more and have a greater appreciation for the field of neuroscience research and the amazing things we're able to do. But if you have any questions, I understand this is a very complicated topic. So if there is anything you would like to ask, anything I can clarify for you, I can do that now.
I have a question. You were talking about doing research on fear-based memories.
Yes.
So what was the research done on? Was it done on an animal?
Yes, we were performing research on rats. Rats, as it turns out, it doesn't seem like it, but their central nervous system, meaning the brain, is actually very similar to the human nervous system. So by investigating rats, we were able to extrapolate that information to humans.
And one more question. How do you get to those memory cells in the brain of that rat?
Yes. Unfortunately, you do have to kill the rat. Basically, 5 minutes after you activate the last memory that you want to investigate, within those 5 minutes you have to kill the rat, which we do very humanely, I assure you, and then remove the brain and freeze it, and then you can slice the brain into very thin slices with a machine called the cryostat. It's like a very expensive deli slicer, and you get these little sheets of brain that you can put onto a microscope.
Okay. That's all the questions I have. Thank you.
All right.